Hello, and welcome to our FDA Animal Biotechnology Case Study webinar. My name is Dr. Heather Lombardi, and I'm the Director of the Division of Animal Bioengineering and Cellular Therapy at FDA Center for Veterinary Medicine. This webinar is part of a series of ongoing outreach initiatives to provide helpful information to our stakeholders and to enhance the predictability, transparency, and efficiency of our animal biotechnology product review process. Today's case study is intended to provide an understanding of the data and information FDA evaluated to support its low-risk determination, announced on March 7, 2022, for an intentional genomic alteration, or IGA, in genome-edited PRLR-SLICK cattle. PRLR-SLICK cattle is enrolled in FDA's Veterinary Innovation Program, or VIP, which was developed to advance innovative animal products by providing greater certainty in the regulatory process, encouraging development and research, and supporting an efficient and predictable pathway to market for IGAs in animals. Under VIP, a SLICK cattle review team was formed of subject matter experts from the divisions of Animal Bioengineering and Cellular Therapies, Human Food Safety, Scientific Support, and Compliance. Review team members will be presenting an overview of their risk assessment today. We will also provide information on FDA's science and risk-based oversight of IGAs in animals, where the level of FDA review and data expectations are commensurate with the risk profile of the product. We welcome and value your input and look forward to hearing from you. We will be providing our contact information at the conclusion of the webinar for you to provide your feedback or to ask us any questions. It is my pleasure now to introduce Dr. Adam Moyer, team leader of the Animal Biotechnology team, who will be providing an overview of FDA's Animal Biotechnology risk-based review process.
Thank you. This presentation will provide an overview of FDA's risk-based review process for IGAs, alterations made using molecular technologies that introduce changes to the genome of an animal. Specifically, we will go over a case study discussing FDA's low-risk determination announced on March 7, 2022 for marketing of products, including food, from 2 genome-edited beef cattle and their offspring after review of data, which demonstrated that the IGA is low risk and does not raise any safety concerns. This decision regarding the IGA resulting in a short, slick hair coat in cattle, shown in the photo and the animal on the left, is the agency's first enforcement discretion for an IGA in animals for food use, but we have previously made many other low-risk determinations for IGAs in animals, including for glowing aquarium fish and animal models of disease. FDA uses this risk and science-based review process focusing on safety to the animals containing the IGAs, safety to people or animals that consume food from the animals, and risk to the environment. FDA uses a risk-based review process, meaning that the scope of the review process is based on the product's risk profile. For certain IGAs, such as IGAs in laboratory rodents used for research, FDA does not review data prior to marketing. For others, such as the IGA presented in today's case study, FDA reviews data and information to confirm the IGA in animals is low risk prior to marketing. In these scenarios, FDA does not expect the developer of the IGA to pursue the FDA's approval prior to marketing, also known as enforcement discretion. For products that are not considered low risk, such as IGAs with human health claims or that are intended to produce a human therapeutic, FDA does expect the developer to submit an approval application. FDA determines risk on a case-by-case basis, and FDA's evaluations consider both the qualities of the specific product that is the subject of the request and its intended use. The example provided here today is based on the information provided by the developer in support of a low-risk determination for PRLR-SLICK cattle. The data we looked at in this case may not be the same for another product. However, the general principles still apply. For example, this developer chose to characterize the IGA and unintended alterations using whole genome sequencing. Another developer of a similar type of product may choose to use a targeted sequencing approach to answer the same safety questions. The risk assessment review process for this case study is shown here. The developer began by contacting CVM early in development. At that stage, they described their IGA and its intended use, and CVM used that information to determine that a request for review of data in support of a low-risk determination was appropriate. CVM informed the developer of that decision, who then in turn opened a veterinary master file, or VMF, a confidential file type that is not subject to user fees. CVM and the developer then met to discuss the data expectations for the request before the developer submitted such data and information to demonstrate low risk. CVM then conducted a risk assessment using the submitted information and any other relevant information that was identified to assess risk to animals, humans, and the environment. As CVM conducted the risk assessment, the developer also listed their IGA and establishment registered to comply with 21 CFR Part 207. Listing and establishment registration helps FDA maintain a current inventory of regulated products and identify where manufacturing operations are performed in case any issues that affect product safety or quality are identified once the product is marketed. Once the risk assessment was complete, CVM issued a letter outlining the decision. This letter described FDA's decision to not object to marketing of the product without an approval application or enforcement discretion based on a determination of low risk. FDA publicly announced its decision and published a risk assessment summary outlining the types of data and information we reviewed and our conclusions. The developer gave CVM permission to share the risk assessment summary and had the opportunity to review the document to confirm that it does not include any confidential information. A link to the risk assessment summary is provided at the end of the presentation. The risk assessment we used here involved a collaborative review team process. The developer directly interacted with the review team, which was composed of subject matter experts in molecular biology, bioinformatics, surveillance and compliance, animal safety, human food safety, animal food and feed safety, and environmental risk. Questions arising from either the developer or members of FDA's review team were communicated and discussed throughout the review process. To discuss more specifics about the review of genome-edited PRLR-SLICK cattle, I will now turn things over to Dr. Stella Lee, molecular biologist and the SLICK cattle review team lead.
Thanks, Adam. The risk assessment we will walk you through today is specific to the IGA in PRL-SLICK cattle. We will discuss the risk assessment, focusing on the risk questions FDA asked during the review process and types of data and information FDA evaluated to make the low risk determination for the IGA in PRL-SLICK cattle. FDA's risk-based review process is product specific, so we encourage early interactions with the developers to discuss data expectations for developers' specific product and requests and potential alternative approaches to address risk questions where it's appropriate. The subject of today's case study is an IGA that is equivalent to naturally occurring mutations that exist in conventionally bred cattle. The intended IGA mimics naturally occurring SLICK mutations by disrupting the bovine prolactin receptor gene, or PRLR, resulting in truncated PRL protein and short SLICK hair coat in cattle. After reviewing the developer's inquiry, CVM determined review of data addressing relevant risk factors demonstrating low risk prior to marketing is the appropriate regulatory approach. In this case, we discussed with the developer that their submission should include data information to address the following key questions. Is the IGA in PRL-SLICK cattle equivalent to SLICK mutations that exist in conventionally bred cattle? For instance, do the animals with the IGA have an equivalent genotype to animals with naturally occurring SLICK mutations? And do the PRL-SLICK cattle exhibit the same SLICK phenotype as observed in conventionally bred cattle? Is there a history of safe use in animal agriculture food production, meaning a history of safe consumption of food from animals and no known animal safety concerns? As mentioned before, the review process used a collaborative multidisciplinary approach. We, the review team, will now walk you through the risk questions, the types of data information we evaluated, as well as our conclusions for the IGA and PRL-SLICK cattle. I'll start off discussing molecular characterization. Molecular characterization serves as the foundation of the risk assessment and involves identifying hazards or potentially risks to humans, animals, and the environment through molecular data. Such data information can be leveraged to support other aspects, and that was the case here as well. Molecular data information was leveraged to support assessment of potential risks to the target animal, food safety, and the environment. To determine whether the IGA contains a sequence equivalent to one found in conventionally bred cattle and whether there are any safety concerns, we evaluated the following molecular data information. First, experimental design and methodology used to create the IGA. Second, the current literature describing the naturally occurring swing mutations. Third, the genotypic data, in this case, whole genome sequencing data, the developer chose to generate to characterize the IGA and any unintended alterations in PRL-SLICK cattle. So first, we evaluated the details of the experimental design and methodology used to create the IGA in PRL-SLICK cattle. Understanding the details of the experimental design would help us to address what potential hazards are associated with introducing the IGA in animals, and to assess if the IgA itself and the bovine PRLR gene and any components of the methodology pose potential risk to humans, animals, or the environment. In this case, briefly, the IgA was introduced by genome editing, specifically CRISPR-Cas9. The CRISPR reagents were microinjected into the embryos prepared by in vitro fertilization. The edited embryos were then transferred into surrogates, and edited calves were born. As we will talk more in the next few slides, data supporting molecular characterization of the IgA in PRL-SLICK cattle was based on a total of 4 beef cows. We evaluated information such as the components of the genome editing complex, their sources, and how they were prepared. For instance, the target site in the bovine genome, the guide RNA sequence, for instance, the homology, if there's homology elsewhere in the genome, as well as the sequences of the intended IGA. Also, we reviewed the type of Cas9 and its delivery method to assess the nuclease specificity and likelihood of unintended on and off target effects. Second, we reviewed current literature describing the naturally occurring swing mutations identified in conventionally bred cattle to compare them with the IGA in PRL-SLICK cattle. According to the literature, there are several different SLICK mutations that exist stably in populations of multiple breeds that adapted to tropical environments. Mutations are autosomal dominant. In other words, an animal with a single copy of such mutation exhibits the SLICK phenotype. While the genomic sequences of these mutations vary, the SLICK mutations identified to date in populations of multiple breeds are located within a certain genomic region, located in the portion of the PRLR gene encoding the intracellular domain of the protein. They contain premature stop codons, resulting in PRL protein truncation, in particular loss of C terminus from the long isoform. PRLR protein truncations occur after certain tyrosine residues and before another conserved tyrosine residue, resulting in the same reduced number of encoded tyrosine amino acids, as you can see in the diagram here. Please note that the diagram is not to scale and only showing tyrosine residues at the intracellular domain. So while the molecular mechanism by which SLICK mutations exert their effects is currently unknown, tyrosine residues of prolactin receptor are thought to be critical for its function. Even though the naturally occurring SLICK mutations result in truncated prolactin receptor of varying lengths, the current literature suggests that they are functionally equivalent, given that the mutations result in the same reduced number of encoded tyrosine amino acids, and importantly, result in the same SLICK phenotype. Thus, based on the current literature and information about the various SLICK mutations identified to date, we consider the IGA to be equivalent to the naturally occurring SLICK mutations observed in conventionally bred cattle if it contains premature stop codons predicted to result in prolactin receptor protein truncation within the same amino acid range as observed in conventionally bred cattle. Next, to discuss the genomic data we evaluated to confirm the IGA sequence and characterize unintended alterations in PRLR-SLICK cattle, I'll now hand it over to Dr. Alexis Norris, the review team's bioinformatician.
Thank you, Stella. For the molecular characterization, the developers submitted whole genome sequencing data, or WGS for short, for the 4 calves and their unedited parents. In addition to evaluating the developer's results, we performed an independent bioinformatics analysis of the raw WGS data as follows. We first aligned the whole genome sequencing reads to the bovine reference genome. We then identified variants that were present in the edited calves, but not in their unedited parents. In doing this, we looked for both short variants, such as short nucleotide polymorphism and short insertions and deletions, as well as large alterations, including deletions and duplications. Importantly, we also looked for very low-level alterations, and this was to account for possible mosaicism. We then used genome annotation data to guide the risk assessment of the identified variants in the edited calves, but not their unedited parents. The genome annotation included repetitive elements, genes, natural variation, and conservation scores. Both the developer's analysis and our own independent analysis found that the cattle with the IgA have a genotype equivalent to mutations found in conventionally bred cattle. Of the 4 calves, 3 had an IgA in the prolactin receptor gene, here represented as the blue animals, while the one unedited animal is here represented in white, and we also call this animal the no-take. While the specific IgA sequences varied in each calf, they were all predicted to result in a prolactin receptor protein truncation that's analogous to naturally occurring mutations observed in conventionally bred cattle. One of the 3 edited calves died due to bovine congestive heart failure, and here that's represented as the animal with a red strikeout. But this could be attributed to genetic markers called SNPs that were inherited from its unedited parents, suggesting that the SNPs were not newly acquired during the process of introducing the IgA, and that the calf's IgA was unrelated to its bovine congestive heart failure. Next slide, please. Both the developer's analysis and our own independent analysis found that the cattle with the IgA are mosaic. This means that in different cells or tissues of the cattle, different alleles may be present. The mosaicism was due to the method used to introduce the IgA into the embryo. There is a statement on the IgA label that notes the presence of mosaicism and provides information to customers, creating the first generation animals from the PRLR-SLICK genetics. Both the developer's analysis and our own independent analysis identified unintended alterations, but these alterations were determined to not pose safety concerns. From available genome annotation data, there is no expected impact on protein expression based on the types and locations of these unintended alterations. One of the unintended alterations is a duplication located in a repetitive energetic region, while the other unintended alterations are all short insertions or deletions that range from 1 to 11 base pairs that are located in either intronic or intergenic regions of the bovine genome. Also supporting the conclusion that the unintended alterations do not pose safety concerns are data supporting that the cattle appear healthy based on visual inspection. To discuss the animal health data evaluation in more detail, I'll now hand it over to Dr. Harlan Howard, the review team's animal scientist.
Thanks, Alexis. The intended SLICK hair coat phenotype was present in 3 of 4 animals. The normal hair coat animal did not contain the IgA. Although thermotolerance was not evaluated with these cattle, it's reasonable to expect that the SLICK hair coat will impart the thermotolerance trait based on published reports on the association of the SLICK hair coat and improved thermotolerance in cattle that have the naturally occurring mutations to the prolactin receptor. Review of genotypic characterization data illustrated that the introduced IgA was analogous to naturally occurring mutations from conventionally bred cattle with no identified unique risks to animal health. Although the amount of animal health and safety data available was limited, both with respect to the number of animals and the types of observations available data provided on routine health observations, veterinary examinations, and live weight gain, coupled with the application of typical management practices for conventional cattle, indicated that the health status of cattle with the IGA was comparable to conventionally bred beef cattle. One calf died with a diagnosis of bovine congestive heart failure. From genetic analyses, referred to the previous discussion on molecular characterization, the condition was genetic in nature, traced back to the animal's parents, and was unrelated to the IGA. To discuss our human food safety evaluation for this product, I will now turn things over to Dr. Kimon Kanalakis, toxicologist from the Division of Human Food Safety.
Thank you, Harlan. During the review of the food safety, there were two main questions. First, we evaluated if the IGA may be present and result in any hazards to consumers from consumption of food derived from the PRLR-SLICK cattle. Second, we asked if there was a history of safe consumption of beef from conventionally raised cattle with the same genotype and phenotype that would also support that the risk to consumers is low. Data from the genotypic and phenotypic characterization reviews were used to identify human food safety hazards. For example, do certain observations on animal health translate to food consumption risk? Based on the bioinformatic analysis of the whole genome sequencing data from the PRLR-SLICK cattle, no hazards were identified. The IGA is consistent with mutations that are seen in conventionally bred cattle. Comparable phenotype and health status were shown in PRLR-SLICK cattle to animals that have a naturally occurring SLICK phenotype, and no identifiable direct or indirect effects on food safety from the edits to the prolactin receptor gene or from the IGA were found. Based on this information, no changes to the composition of the food from PRLR-SLICK cattle was expected. Therefore, we conclude that we have met the standard of reasonable certainty of no harm to people from consumption of edible tissues from PRLR-SLICK cattle. This is the same food safety standard used for approving an IGA in an animal. Additionally, the Division of Animal Food Ingredients conducted an evaluation and determined that there were no hazards identified that would result in a safety concern for rendered PRLR-SLICK cattle used in animal food. Next, Rebecca Cluster from the environmental team will be providing an overview of the environmental risk evaluation.
Thank you. Although the decision not to enforce the approval requirements based on low risk is not considered a major agency action under the National Environmental Policy Act, we did evaluate the risk to the environment from the marketing of the IGA in PRLR-SLICK cattle and their associated products. Prior to deciding if we would expect submission of an approval application for the IGA in these cattle, we wanted to know if the risk to the environment was low. The environmental risk assessment first aims to identify potential hazards to the environment resulting from the IGA, and then analyzes the potential risk to the environment from those hazards, as well as the risk to the environment from the animal that contains the IGA. No environmental hazards were identified with regard to the IGA based on the genotypic data submitted by the developer. Specifically, no new expression products are expected. The IGA is intended to produce a truncated PRLR protein, which already exists in cattle populations throughout the world. In addition, the phenotype produced by the IGA is a well-established trait in several breeds of domestic cattle that are currently produced in the US. As for the scope of the decision, these PRLR-SLICK cattle can be housed on any farm in the United States. The likelihood of establishing a population of PRLR-SLICK cattle in the US environment is low. Upon escape or intentional release, there are very few feral cattle populations with which the animals could breed. Cattle are also fairly easy to recapture due to domestication and their large size. In addition, animal carcasses and waste can be disposed of according to typical farm practices. No hazards were identified with the IGA, and the trait is already present in US cattle populations. Therefore, the manure and carcasses would be no different from conventionally raised domestic cattle. Based on this information, we concluded that the risk to the environment resulting from the marketing of the IGA contained in the PRLR-SLICK cattle is low. Now I'll turn it over to Dr. Emily Cornwell, a veterinarian in CVM's Office of Surveillance and Compliance, who will discuss the review team's risk determination.
Thanks, Becca. After reviewing all the information discussed earlier in the presentation, FDA determined that the IgA and PRLR-SLICK cattle poses low risk to humans, animals, and the environment. To summarize, during the risk assessment review process, we determined that the genotype in these cattle was equivalent to naturally occurring SLICK mutations found in conventionally bred cattle, recognizing the normal biological variability that was outlined in the molecular characterization evaluation. We also determined that these cattle have the same SLICK phenotype for which there is a history of safe use in animal agricultural food production. We did not identify any human food safety concerns during the review, and an environmental review determined this product posed low risk to the environment. As a result of this determination, FDA does not expect the developer to submit an application for approval for the IgA in the SLICK cattle prior to marketing the cattle or its associated products, for example, offspring, semen, embryos, or meat. This decision is limited to the products derived from the existing 2 cattle containing the IgA for which FDA has reviewed data and their progeny. If the developer wishes to broaden this determination, they may submit additional data to support a broader determination in the future. As mentioned in an earlier slide, the listing and establishment registration requirements in 21 CFR Part 207 apply to IgA products. In this case, FDA intends to treat facilities or farms that are engaged in standard agricultural practices for PRLR-SLICK cattle, including assisted reproduction techniques such as embryo transfer or raising the cattle for food production, the same as facilities or farms that are engaged in these practices for cattle without IgAs. In other words, FDA does not expect facilities performing standard breeding practices for these cattle to register or list. Whether these requirements apply to a particular product is something we will consider based on the facts for that product. This slide provides links to some additional resources that may be helpful, including the risk assessment summary for the PRLR-SLICK cattle and information on the Veterinary Innovation Program. These resources are all available on our website, fda.gov. We welcome any feedback and questions you may have on this webinar. Please submit these to fda-cvm-animal-biotechnology-feedback@fda.hhs.gov. This slide contains additional contact information you may find helpful, including the email address for general inquiries, askcvm@fda.hhs.gov, the email address for biotechnology inquiries specifically, askcvm-biotech@fda.hhs.gov, and Dr. Lombardi and Dr. Moyer's email addresses if you wish to contact them directly. Thank you for your attention. We hope you found the information in this webinar to be helpful.